Hello, everybody. Welcome to Ms. Maguire online video lecture. Today, we continue covering introduction to stem cells. In the first video, we talk about stem cells, what they are, how we can classify them. And I just want to remind you that we can classify stem cells based on their potential, right? They can be totipotent, pluripotent, multipotent, or based on the source that we use to collect those stem cells. We can have embryonic stem cells, adult stem cells, and induced pluripotent stem cells. All of them have their own advantages and disadvantages, and please watch the first part of this video lecture to learn about learn this general information about the stem cells. But today we're gonna start with a slide that kind of where we stopped last time, and we will talk about cloning. So I'm trying to find the slide. Okay, so here. Now, what is cloning? Cloning is the growing of organ or entire organism from a single cell, right? So if you have a stem cells and we want to make organs, right? So we can grow organs or tissues, and we want to use this tissue to replace the damaged tissues or organs of a patient. So we need to apply this cloning to stem cells concept, right? So let's talk about different type of cloning first, right? So there are two very different types of cloning. Reproductive cloning. Let me pick up my pen. And this is when we make two identical individuals. It's very difficult to do, and it's illegal to do to human. And there is molecular cloning. We use molecular cloning to study what gene does, and it's used very often in a biological lab. So it's used routinely in biological labs. And molecular cloning is when we make recombinant DNA. So we can take a gene from one individual. So it's shown here in the red. We cut it out and we will insert it into DNA of another individual. So we form the recombinant DNA. And so that's a molecular cloning. So reproductive cloning, a cell from the body is used. Then we will remove the nucleus from the cell, right? And we know that nucleus has all the DNA. And we use an egg. We remove nucleus from the egg. So egg used like a pretty much container. Remember, the human egg or ovum has all the organelles that we need. It has ribosome, RAF-ER, Golgi apparatus, right? And it has its own DNA. But we will get rid of this DNA. We're gonna insert the DNA from a body of this sheep, for example, into an egg, and and then we fertilize it. Oh, well, we don't need to fertilize it because this nucleus already is to egg, so it's already diploid. In the egg, remember, we have only half of the chromosome, so it's one N. So we have in human egg, we have only 23 chromosomes. So that's why we need sperm to fertilize. In this situation, we take nucleus from somatic cell. So that nucleus already has 46 chromosomes if we're talking about humans. I don't know how many chromosomes sheep have, honestly. But human nucleus have 46 chromosomes, so we don't need any sperm. So we have the zygote, right? We skip this process of fertilization. So if you have a zygote, the zygote will divide, and we produce identical organism. identical to the ones that we get nucleus from. So this is how the first organism was produced. It was a sheep named Dolly, and it was the first clone of already existing animal, right? Dolly unfortunately didn't live long enough, but it was pretty successful experiment. Okay, so that's our, this one is reproductive cloning. And here's our molecular cloning. We will take DNA out of the nucleus. Then we will make a new piece of DNA. And we will put new DNA into a test cells and grow copies of it, right? So here's cell number 1. It has, DNA, and there is a particular gene. So here we have cell number 2, and it has a different DNA. So, maybe this is the DNA of a strawberry, and this is DNA of a fish. Two completely different organisms, DNA of strawberry and DNA of fish. So maybe we will take this gene from a DNA of a fish, we cut it out, and we will insert it into DNA of strawberry, right? Then we're gonna insert this DNA inside a cell, strawberry cell maybe, and we will grow transgender organism. So in this situation, daughter cells contains DNA from 2 different organisms. Gene 1 from strawberry, for example, and gene 2 from a fish. And we can clone it, so it can, cell will divide and grow, and we will have transgenic organism. Now, these experiments are done routinely in a lab, so we can do this. It's easy, it's not that expensive, and it doesn't involve really, usually it's used to study function of genes or some drugs, how drugs work. We don't use it to produce animals. We do, however, use this technology to make plants with recombinant DNA. Right? Okay. We use this cloning in therapeutical researches, and for to make maybe some tissues or cells, maybe individual cells that we can use to help people to deal with their diseases. Now, what is therapeutic cloning? DNA is extracted from a human cells. So this DNA is then inserted into a woman's ovum and develop and produce stem cells, right? So instead of kind of collecting these stem cells, we can produce them if we use DNA from somatic cells and insert it into an egg of a female. Stem cells are removed from pre-embryo, and they're grown in a specific organ and then can be transplanted into the patient, right? So that's our therapeutic cloning. So this is how it works. So here's the egg, and nucleus is removed. Now we have this patient. We take a somatic cell of a patient, so it's a normal body cells, but we're not using the cell. We're only using a nucleus from this patient cells. It's transported inside the egg, and we have fertilized egg. And we start growing this egg. So it will divide, forming the ball of cells and blastocyst. It's a hollow ball. And then we will remove those cells of inner mass. Remember, those cells are pluripotent cells, pluripotent embryonic cells. And that's our stem cells. So we'll have it in the media. We will grow pluripotent stem cells that can differentiate into skin cells, nerve cells, or pancreatic cells, or other cells of your body. And then we will use these cells to help this patient to deal with a disease or some genetic conditions, right? So we can grow the stem cells of this particular patient. So for example, it has a nerve damage, right? And nerve cells are already differentiated cells. They cannot divide. So why not to use stem cells to grow new nerve cells, right? And help this patient to heal after spinal cord injury, for example, or injury of some peripheral nerves, right? So that's how stem cells can be used. And we use a cloning over here, right? Because cloning is growing of, in this example, stem cells from patient somatic cells using an egg with a removed nucleus. Right, so here's a promises of stem cell research. Cultured pluripotent stem cells can be used to study cell differentiation and understanding and prevention and treatment of birth defects. It can be used for identification of drug targets and test potential therapeutical therapeutics, like some drugs, how drugs affect our cells. Stem cells can be used for testing of toxins, toxicity testing, and of course for tissue, cells, and possibly in future organs for transplantation, because those stem cells can grow into bone marrow cells for leukemia and chemotherapy, in the nerve cells for Parkinson, Alzheimer's disease, heart muscle cells for heart disease, or pancreatic islet cells for diabetes. And of course, we have this square here of unknown. What are the other areas we can use the stem cells? And to finish up this chapter, let's talk about cancer stem cells. Cancer stem cells are defined as those cells within a tumor that can self-renew and drive tumorigenesis. So remember, characteristic of all stem cells is that they can make more stem cells. It's called self-renew. And plus, stem cells, they divide, right, into and differentiate into other cells. In this situation, cancer stem cells can differentiate into other cancer cells. Now, cancer cells have been isolated from a number of human tumors, including hematopoietic, that's a cancer of blood, brain cancer, colon, and breast cancers. The cancer stem cells concept has important implication for cancer therapy. So let's compare cancer cell with a regular stem cell. So here is the, oh, okay, interesting. Oh. I don't know I can do that. So here's our, oh, that's, I'm sorry. Something new to learn every time. So here's our regular stem cells, normal stem cells that they self-renew. That means they can make more stem cells, right? So hopefully we will never run out of them. And at the same time, they can differentiate into a committed progenitor. And those committed progenitor then differentiate into terminally differentiated cells. So those are our like muscle cells or nerve cells, right? And they not dividing cells. And the stem cells, as shown over here, differentiated into committed progenitor cells. So the point is the stem cell, they divide and make more cells, more stem cells and make progenitor cells. Then they differentiate into terminal cells, mature cells that do not divide anymore. However, in a cancer cell, we have cancer cells will renew, they will form more cancer stem cells, and they will form cancer progenitor cells. And those progenitor cells will differentiate into cells of the cancer, but the point is they're still dividing, right? So they're still dividing cells. So there is a similarities, right? And at the same time, if it's a normal stem cells, those mature cells, they do not divide. In a cancer case, the cells still dividing. Even differentiated cells are still dividing. So that's how it, this is how it looks when we talk about tumor. But first, let's get some more information about these cancer stem cells. They are really minor population in tumor. It's only 0.1% in a tumor of these cancer cells. So here is shown a tumor, and some cells over here are cancer stem cells. They can self-renew as all stem cells. It means infinite proliferative potential. So that means they can divide without any limitations. In addition, they have enhanced resistance to drugs, radiation, and cell stress. They're tumorigenic. They give rise to other cell type in tumors, and they associated with metastasis and relapse. Relapse means you think you cured from cancer and then it's reappeared. Like, why? So here's the tumor of cancer cells, and we use some anti-cancer drugs or radiation, right? And we kill the tumor cells, so we completed the task. However, cancer stem cells, they highly resistant, so they survive. And because they tumorigenic, they grow into tumor again. And this way we have relapse of the tumor. Even, we have, for example, somebody with breast cancer, and they went through their radiation therapy, through the surgery, they removed all the cancer cells. They hope that they destroy the tumor, and they might. However, some cancer stem cells can still survive. Not only they can grow back into tumor, but they also can migrate to a different part of the body. So metastasis and relapse are involved in more than 90% of all cancer death. So people die from cancer, 90% of people die from cancer because either relapse, so what we say cancer came back, or metastasis, where these cancer stem cells spread to a different part of the body. So strategies to eradicate cancer stem cells are an urgent topic in cancer research. So just, we know how important stem cells are in the development of new drugs or organ transplants, or even to replace cells, for example, pancreatic cells, beta cells that produce insulin. If we have people with diabetes, those cells do not function properly. So what if we can have stem cells differentiate into normal pancreatic cells and replace the ones that is not functioning? But there is also devastating diseases like cancer, for example, right, that use these cancer stem cells to continue spreading through patient's body, right? But we still want to finish up on a good note. So today's stem cells-based therapy has mainly focused on leukemia and other cancers, diabetes, Parkinson, Alzheimer, spinal cord injury, and heart diseases. So this is the area where we can hopefully successfully use the stem cell technology to help defeat those devastating diseases. And we use stem cells tissue engineering, when we can grow tissue and replace the tissue that is damaged. And here are some successful results shown here. We can grow skin from stem cells, bladder, muscle, bone, cartilage, and nerve. Okay, so still we still don't fully understand what stem cells are and how we can use them for our advantage. But it's interesting and promising research, and hopefully it will make difference in our lives and lives of our children and grandchildren. Okay, and the last slide I want to show you, I want to give a credit to people who created some of the slides, and I borrow their work to make my presentation. So that's it for today's lecture. Thank you very much for watching, and I hope it was helpful.